Hello everyone. In this video, we'll talk about how we classify living things and how humans interact with life. Keep in mind, I will only be covering things studied under microbiology. So what is microbiology? Microbiology is a branch of science that deals with microorganisms such as protozoa, fungi, viruses, and bacteria. These microorganisms can be very beneficial to us. Inside of the body, we have microorganisms that help us with digestion. It's also helpful for nitrogen fixation in plants and breaking down cellulose for digestion in animals. We divide bacteria, viruses, and fungi according to their characteristics. With bacteria, we use a technique called gram staining to identify them, usually used for choosing the best antibiotic for a person. In viruses, we depend on their genome. And in fungi, they're either divided into molds, which are multicellular, or yeasts, which are unicellular. The Linean Hierarchy developed by Carl Linnaeus consists of several levels that go like this: domain, kingdom, phylum, class, order, family, genus, and species. Starting from the top of the list, we have 3 domains: bacteria, archaea, and eukarya. Bacteria and archaea are considered prokaryotes, but eukarya are considered as eukaryotes. Prokaryotes are small unicellular organisms that don't have nucleus or membrane-attached organelles, while eukaryotes are multicellular organisms that do have membrane-attached organelles. After that, we go to the kingdom. Bacteria and archaea have only one single kingdom, which are bacteria and archaea respectively. The eukarya has 4 kingdoms: protists, fungi, plants, and animals. And then we continue the classification as mentioned before. For now we will focus on bacteria. How do we classify them? How does gram staining work? Any organism's name is composed of 2 words. The first name is the genus, the second name is the species. The first letter of the genus is always capitalized, and both are written in italics. This is called binomial nomenclature. Bacteria associate in different arrangements. Their names are given prefixes based on how they attach and their shapes. Coccus means round shape, bacillus means rod shape, and spirillum means spiral shape. Diplo means in pairs, strepto means in chains, and staphylo means in clumps. For example, streptococcus would look like round shapes arranged in chains. Moving on, prokaryotic bacteria cells have the following: a tail-like organ called the flagellum, which is used to move; nucleoid, where genetic material is located; ribosomes, responsible for protein synthesis; the pilus, which attaches to another cell to perform conjugation; plasmid, which is an extra chromosome of genetic material, so it's not part of the DNA; capsule, which is a protective layer; and cell membrane, cell wall, and cytoplasm. Now, moving on to gram staining. In gram staining, we can classify bacteria into either gram-positive or gram-negative, and that depends on the cell wall constituents and its ability to react with the gram stain. To classify them, crystal violet dye that targets the peptidoglycan in the bacteria's cell wall turns it purple. Then, safranin dye is added to turn the remaining cells pink. Therefore, gram-positive bacteria have a thick peptidoglycan cell wall and turn purple, while gram-negative bacteria have a thin peptidoglycan cell wall and turn pink. That's about it. Thank you for watching.